Hi, my name is Marjory Moreau. I am the Associate Director of the Computational Toxicology Division at Cytovation, and today I'm going to make a presentation on the use of PBPK modeling in vitro to in vivo extrapolation, or IVIVE. So the new paradigm is to move from traditional animal experiment to new approach based on a combination of in vitro and in silico methods. The high cost of in vivo testing in terms of time, resource consumptions, and animal usage has confined its usage to testing limited numbers of compounds in a low throughput manner. So we need alternatives, strategies for chemical risk assessment. The interpretation of high throughput screening data and other in vitro methods require in vitro to in vivo extrapolation, IVIVE, for interpretation in a risk assessment context. So how to interpret these in vivo data? Can we link in vitro exposure with in vivo exposure? And what key relationship can we use to help interpret the data? So with the new toxicity paradigm and the increased use of NAMs, there has recently been greater recognition of the use of computational models like pharmacokinetic modeling and IVIVE to help interpret biological data and integrate various types of studies that can eventually be applied in a risk assessment context. So IVIVE is really a valuable tool in extrapolating an in vitro point of departure. So here, a point from an in vitro toxicity assay to an equivalent applied dose in vivo. So basically, chemical concentration here active in an in vitro system is converted to an equivalent level in vivo by using a pharmacokinetic model. So what are the applications of IVIV? So IVIV has been mostly developed and applied in the pharmaceutical industry to estimate therapeutic blood concentrations for specific candidate drugs, and also to identify potential drug-drug interactions. IVIV is now being increasingly used to predict the in vivo pharmacokinetic behavior of environmental and industrial chemicals. So simple PK model can be used for IVIV, and they are really useful because they are pretty simple. They can be rapidly parameterized with just using in vitro measurement of clearance and plasma protein binding. But with additional data, we can use more elaborate model like PBPK model to do IVIV. HCTK and Platham, for example, have PBPK models to generate equivalent dose for batch of chemicals. So here it's an example of the use of a simple PK model. We can use them to prioritize compound using in vitro bioactivity data and exposure estimate. So here in this example that is taken from Ring and al, you can see on the top here, we have the results of in vitro high throughput screening assay plus high throughput IVIVE to estimate a dose to cause bioactivity. And on the bottom here, we have high throughput prediction of exposure. And here we can calculate an activity exposure ratio, which is kind of a margin of exposure, and see the decreasing priority of the chemical. So the larger the margin, the better. So PBPK modeling are really useful because we can incorporate kinetics in the process. So we know that in vivo toxicity is affected by pharmacokinetic factors, but they are not reflected in vitro toxicity tests. So data on absorption, distribution, metabolism, and elimination can be generated using in vitro and in silico methods. And PBPK modeling will serve as an integration and translation tool for IVIVE. So how does it work? So physiological structure is well characterized for PBPK model. I've created another session explaining how PBPK models are created and used, but basically everything related to tissue volume, blood flows, ventilation rates, body weight are all well-known parameters that can be found in the literature. For chemical-specific parameters, they can be derived from, obviously, in vivo, if there's some in vivo data, but now with the need to reduce animal experiments, they can be derived from in vitro and in silico methods. There's other data that are needed for parameterization. So, for example, blood-air partition coefficient that can be predicted. Urine clearance and GFR for water-soluble compound, binding to plasma protein and red blood cells is also really important. And for oral absorption, the intestinal absorption is also important. Something that also needs to be taken into consideration is the fact that in the system itself, the in vitro system, there's some binding or other mechanism happening that can limit the availability of the compound. So in vitro, free concentration may only be a fraction of the nominal or totally added concentration because the chemical may evaporate, bind to serum constituent or well-placed plastic, for example, and this will reduce the concentration available for uptake into the cells. So it's really important to take this into consideration. So what is IVIVE? So IVIVE is an integral component in new approach methods for rapidly translating in vitro toxicity assay results in the context of in vivo exposure. So IVIVE converts active concentrations from an in vitro assay to a human equivalent dose or human exposure that would be expected to produce internal plasma or target tissue concentration in an exposed person equal to the active concentration from the in vitro study. So the equivalent human exposure can then be compared to an estimated exposure to obtain a margin of exposure or an activity exposure ratio. To develop a quantitative IVIVE model, like in this figure, a measure of hepatic clearance is needed. This value is often obtained from a measurements of in vitro intrinsic clearance that are extrapolated to in vivo clearance rates with the IVIVS, which is in vitro to in vivo scaling model, and also plasma protein binding. So that's the part we can see here. To populate the model, we need clearance, we need protein plasma binding, and we need renal clearance to transform the in vitro intrinsic clearance into in vivo intrinsic clearance. And then we can use the model to convert the in vitro assay to an in vivo human equivalent dose that can be compared to population exposure to derive a margin of exposure. Traditionally, pharmacokinetic models try to relate exposure concentration or external dose to the concentration of the chemical in blood or tissue. This is known as forward dosimetry. Alternately, measured or predicted blood or tissue concentrations of chemicals can be combined with the known pharmacokinetics of this chemical to estimate the external dose that would be required to achieve this measured or predicted internal concentration. And this second approach is what we call reverse dosimetry. Here we have a simple example of quantitative in vitro to in vivo extrapolation for parent chemical toxicity. So we had a list of estrogenic compounds selected through a prioritization workflow based on ToxCast and their predicted metabolites. So we did two things. First, we did some in vitro uterine estrogen response assay in the lab, and we derived AC50 values, and we used IVIVE to convert these AC50 values from this in vitro uterine assay into a human equivalent dose. And the second part of the project was to look at the literature to see if we could find some in vivo data, in vivo heterotrophic assay data, so we could compare with our in vitro assay and see if the in vitro assay is as sensitive as the in vivo assay. So we found some point of departure from the literature. We applied some uncertainty factor to derive a human equivalent dose, and we compared the human equivalent dose from the in vitro assay with the human equivalent dose from the in vivo rat experiments. So that's one way that IVIV can be used. So the optimal way to evaluate the accuracy of the IVIV-derived predictions would be to compare them against values determined in vivo studies. So here in this example, we have several chemicals with the in vivo derived concentration at steady state. And here we have the IVIV results for restrictive and non-restrictive clearance. So here the IVIV model predictions for 7 chemicals, which are 2,4-dichlorophenoxyacetic acid, bisphenol A, cacodylic acid, carbaryl, oxytetracycline dihydrate, PA4A, and triclosan. So these predictions were comparable to the concentration at steady state values derived from published model. So you can see carbaryl, for example, you can see the values are really similar. Bisphenol A, it's also in the same order of magnitude, which this one also same thing. But out of the 4 remaining chemicals, the IVIV model significantly over-predicted the concentration at steady state. So the assumptions made in this IVIVE modeling bias the results toward concentration at steady state over-prediction, because our assumptions are that clearance is limited to hepatic clearance and non-metabolic renal clearance, and also 100% absorption is assumed. So these assumptions are really conservative from a human health standpoint, because lower absorption or additional routes of excretion would result in a lower estimates of the oral equivalent dose required to achieve a specific plasma concentration. So usually restrictive hepatic clearance is assumed and was assumed here in the pharmacokinetic modeling, where the on-off kinetic of protein binding are slow enough to become rate limiting. However, for many chemicals, hepatic clearance is non-restrictive, which means that all chemical free and bound is available for metabolic clearance. So here, when you look at restrictive clearance, you can see that for some chemical, we had some over-prediction. We have the example here of phenyltropium. But when we switch from restrictive to non-restrictive clearance, you can see that the predictions are now in accordance with what is seen in vivo. We have the same thing for picoram. Picoram with restrictive clearance, the value is over-predicted, but when we switch to non-restrictive clearance, then the predictions are pretty good. Usually at low concentrations, the kinetics are linear. So that's why we can use a simple pharmacokinetic model to derive human equivalent oral exposure. So for that, we need the dose rate times the body weight divided by the total clearance, so hepatic clearance and renal clearance. And then we can use reverse dosimetry. So usually we assume an exposure of 1 milligram per kilogram a day. And we calculate what's the concentration at steady state, and then we can calculate the estimated oral exposure based on the concentration at steady state and the concentration EC50, for example, from an in vitro assay. So here you have an example showing that the simple implementation of IVIV for high throughput assays demonstrates that kinetics is really crucial. So on the left, here, you can see that by themselves, in vitro assay effect concentrations are quantitatively meaningless for risk assessment. But on the right, there is greater than 4 orders of magnitude difference in potencies in vivo. So IVIV is really useful to put into context the results of in vitro assay. You have a simple example of IVIV to predict steady state in vivo kinetics for environmental chemicals. So we can first we derive some in vitro metabolism that we scale through IVIVS to have an estimated in vivo liver intrinsic clearance. We calculate the total clearance by using blood flow and intrinsic clearance. And then we can use a simple kinetic model to estimate blood concentration. So after oral, we use a dose of 1 milligram per kilogram, and we divide by the liver intrinsic clearance and the unbound fraction time the glomerular filtration rate is the renal clearance. For inhalation, it's pretty similar, but we incorporate the air-blood partition coefficient in the equation. And this allows the calculation of the concentration at steady state, which is at the dose that is associated with in vivo effect. And then we can compare with the concentration C in the effect, in the in vitro assay, sorry. IVIV is really useful, but there's a lot of research right now to improve IVIV. Like I've said in a previous slide, we need to consider the free concentration from the cell-based assay, because we need to take into consideration that some chemicals can evaporate or stick to protein or to the constituent of the in vitro assay. We also need to take into consideration, especially when you use a simple pharmacokinetic model, so we need to consider that sometimes the parent is not the toxic entity, but it's the metabolite. But the actual IVIV equation does not take into consideration metabolism. So that is something that is really important and that we need to look into more. We also do not take into consideration extrahepatic clearance, like biliary excretion, for example, or even GI metabolism. So it's really important, and there's some work that still needs to be done to improve IVIV. This slide basically resume what I just said. So we have some limitation, like accumulation and distribution of compound in tissues that are not considered with the simple IVIV using a simple PK equation. Again, this is what I said about the fact that in the in vitro system, we can lose some compound because it's binding to serum or to the plastic on the wells, for example. So this needs to be done and taken into consideration. Obviously, the actual system does not take into account transport or permeability. So this should be taken into consideration. Plus, it's limited to parent compound and not metabolite. But we know that sometimes the parent compound is not the toxic entity. Again, sometimes it's the metabolite. So it's really important to understand this limitation. The example of the parent versus metabolites. So the clearance of the parent results in formation of the active entity or active metabolite. So toxicity would not be observed or overestimated in test cells with low metabolic competence. So the frequent absence of metabolically competent components in vitro system is a well-recognized limitation of IVIV and current assays. And also the lack of consideration of metabolites in IVIV obviously makes the confidence assessment of risk based on NAMs problematic. In practice, it's really not simple to base decisions on active forms of compounds because both the platform for assessing in vitro bioactivity assays and for measuring in vitro metabolism assays may lack the full complement of enzymatic pathways required to activate a test compound and would likely misrepresent the in vivo phenotype. So I really like this figure that was made by one of my previous colleagues, Patrick McMillan. It represents the effects of in vitro assays that lack metabolism on the potential bioactivity of chemicals. So when both parent and metabolites are active, the results expected from in vitro assays are true positive. So here, when the parent compound is active and the metabolite inactive, the results depend on the clearance. If the compound is cleared quickly, no effects will occur, as the compound will not be in the body long enough to have deleterious effects. However, in a cell culture system, we expect to see a positive response, which we call a false positive in this context, as the in vivo bioactivity is expected to be negative. Another example, when the parent compound is inactive and the metabolite is active, the results expected from in vitro screens are false negative. Indeed, most of in vitro systems lack metabolic competency, so the results of the test will be based on the parent compound, which gives a negative results, even if in vivo it's known that the metabolite is expected to cause toxicity. So this is really, really important to take into consideration when doing IVIVE. Now let's look at PBPK models and how we can use them to do IVIVE. So in vitro toxicity assay will give us a point of departure here that we can use in the PBPK model to derive an oral equivalent dose. This is something that can be used to compare species sensitivity, for example. I think the best way to show you how PBPK models can be used to do IVIVE is to show you two case examples. The first one was published recently. So 5 human-derived differentiated in vitro airway epithelial cells culture models were exposed to a non-toxic vapor 1,3-dichloropropene or 1,3-DCP. We monitored the toxicity of these cultures 24 hours after acute exposure using an assay for transepithelial conductance. So it's for the epithelial barrier integrity, and an assay for cytotoxicity. So it's for lactate dehydrogenase or LDH release. Finally, we developed an airway dosimetry computer model for 1,3-DCP vapor to predict in vivo external exposure scenarios that would reproduce toxic local tissue concentration as determined by in vitro experiments. So I really like this figure because it really explains really well for the reverse dosimetry and IVIVE. So with a generic model, you can do two things. If you have in vivo inhaled exposure in species X, you can use a PBPK model for species X and for the chemical of interest, run forward dosimetry, and find out what's the local tissue exposure in our species of interest. Now, if we have tissue concentration, we can use the same model and do reverse dosimetry to find what's the in vivo inhaled exposure that will lead to this concentration in the tissue of interest. Now, application to predict human in vivo response from in vitro point of departure and validation of SS. So that's what we did in the previous slide. We have a dosimetry model in human, and we had some point of departures from in vitro assay in the lung, and we used the model to do reverse dosimetry to predict in vivo inhaled exposure at the point of departure in human. The goal is to compare the predicted human in vivo point of departure to empirical human in vivo NOAL and LOAL for the tissue of interest, if it's available, to see if in silico methods combined with in vitro experiment can replace animal experiment. So here in the transepithelial conductance assay, conductance was increased with 1,3-DCP treatment in a dose-dependent fashion. In the LDH release assay, cell culture models differed in maximal LDH released, when treated with cytotoxic concentration of 1,3-dcP vapor. So mucillar, nasal, small air, and epialveolar released substantially more LDH than did mucillar, bronchial, and tracheal. So for each tissues, we did reverse dosimetry with the PBPK model to calculate the rat equivalent inhaled concentration that would produce after a 4-hour exposure a 1,3-dcP concentration within the lumen adjacent to the tissue of interest that was equivalent to the in vitro point of departure. So in vitro point of departures, which are the blue bars here, and their rat equivalent inhaled concentration in orange are depicted for transepithelial conductance, which is figure A, and LDH release dose response relationships, figure B. The bar heights represents the BMD 1 standard deviation, and error bars extend from the BMDL 1 standard deviation to the BMDU 1 standard deviation. So they represent basically the 95% confidence interval on the BMD 1 standard deviation. Overlaid on this plot are lines here representing the LC50 for rats, so the red line, determined in an acute study from Lomax in 1987, the NOAL here in green, and 2 additional adverse effect levels, so in light and dark orange, from a subchronic study from Stott and al. In 1988. And you can see that the human equivalent dose derived from, the rat equivalent dose derived from the rat in vitro assay are in the same order of magnitude that the result from the subchronic study. So what is the application of this work? So in the case of exposure to a new volatile chemical, so in the panel A, a decision must be made regarding the risk. For example, forward airway dosimetry modeling could be applied to extrapolate that inhaled concentration to luminal concentrations at regions throughout the airway. Those predicted luminal concentration could then be tested in vitro on site-specific differentiated airway epithelial cultures. A finding of acute in vitro toxicity at any of those luminal concentrations would caution against that use of that compound. In the case of multiple volatile compound, so panel B, chemicals can be prioritized to choose the safest option for further development. So a tiered testing approach would efficiently help the decision process. So first, purely computational approaches, such as QSAR modeling, could be applied to reject compounds with a high probability of toxicity. Next, in vitro exposure using an air-liquid interface with affordable immortalized cell lines may be sufficient to reject a subset of compounds with relatively high toxicity. For the remainder, testing in differentiated air-liquid interface cultures, followed by IVIVE to determine a human equivalent dose, would provide more accurate prediction of relative toxicity and allow for further down selections. Lastly, limited in vivo testing could be performed as needed only on the final subset of compound. So now let's look at the example 2, which is combining PBPK modeling and developmental toxicity assays. So in vitro screening platforms to assess teratogenic potential of compounds are emerging rapidly and really show a promising ability to predict the outcomes of in-life animal studies and to provide an attractive means for identifying teratogens and other developmental toxicants. So ReportAcker is a human-induced pluripotent stem cell-based biomarker assay that is shown to reliably identify the teratogenicity potential of new pharmaceuticals and chemicals. These in vitro responses of potential toxic outcomes can be improved for hazard identification applications by introducing a quantitative framework to translate into equivalent human exposures. So basically, the goal of this study here was to evaluate whether the report tracker assay can predict the in vivo developmental toxicity exposure levels of drugs using PBPK model describing their biokinetic behavior and an IVIVE approach. So here we performed IVIVE to predict human equivalent developmental toxicity dose levels of two known human teratogens, thalidomide and carbamazepine, that would correspond with in vitro concentration associated with adverse outcome in a report tracker. So the report tracker is a state-of-the-art human stem cell-based in vitro assay. The assay captures basically the key cellular events of stem cell differentiation in early embryonic development, and it's used to investigate if compounds interfere with early embryonic development, and the assay can also quantitatively predict their teratogenic potential. So here it's the approach we used to do IVIV. We developed a PBPK model, and we ran some Monte Carlo simulations to estimate median 5th and 95th percentiles for the uterus and plasma concentrations for individuals exposed at the same fixed dose of 1 milligram per day. Then we did reverse dosimetry to predict administered dose equivalent to in vitro active concentration from the report tracker assay. For the report tracker assay, we used low L concentrations for both thalidomide and carbamazepine. And both these low L and concentration are assumed to be uterine or plasma concentration, and both scenarios were simulated in this case study. And then comparison with in vivo effects in rat and rabbit could be done. So before doing IVIV, which the results are shown in this table, we did validate the PBPK model. Thalidomide and carbamazepine both have a lot of pharmacokinetic data. So we used published PK literature to validate, calibrate and validate the model. And we also did some sensitivity and uncertainty analysis. All these results, as well as the IVIV results, will be published by the end of the year in a special edition of Toxicology on NAMs. So here you can see you have results for uterus and plasma, because like I said, human equivalent doses were derived using the PBPK model, and assuming that the low L concentration for thalidomide and for carbamazepine from the report tracker are all plasma or uterine concentration. So here you can see that we did run some simulations for bolus exposure or repeat exposure. You have the results here for thalidomide and for carbamazepine. You can see that for thalidomide, the result is the same for bolus or repeated exposure, for the simple reason that in 24 hours, thalidomide is completely excreted, which is not the case for carbamazepine, which is accumulating a little bit. We also did derive human equivalent dose for rat and rabbit low IL that we found in the literature by just applying some allometric scaling based on EPA guidelines. And then we compared the human equivalent dose derived from in vitro experiments from the report tracker in uterus or in plasma with the rat and rabbit human equivalent dose. And as you can see, the human equivalent dose derived from the in vitro assay are in the same order of magnitude than the in vivo experiments. And it's exactly the same for carbamazepine, especially if you look at the repeat exposure, which is probably more accurate to use as a reproductive study is on long term, not acute exposure. So this is really promising. So to conclude, in the report tracker, both chemicals markedly disrupted morphology and decreased the expression of the selected biomarker genes in at least one of the 3 differentiation assays, confirming the teratogenicity of these chemicals in vitro. The ReproTracker-derived human equivalent dose were quantitatively similar to in vivo developmental studies from both rat and rabbits, and indicated that ReproTracker is sensitive and protective of humans. So this pilot study really demonstrated the importance of integrating PBPK model in assessing developmental toxicity in vitro, and also showed that the combination of these tools can be used to improve the safety assessment of drugs and chemicals without animal testing. So this case study is part of a NIHS grant awarded to Cytovation. We are going, like I said, publish a manuscript by the end of the year in a special issue of toxicology on NAMs. And we also started creating a graphical user interface called DRIVE for developmental and reproductive in vitro to in vivo extrapolation. So DRIVE can be used by practicing toxicologists without modeling or programming background. This technology has really a lot of appeal and represents a game changer in toxicology. With DRIVE, toxicologists can screen chemicals for developmental and reproductive effects.